Hello, this is going to be chapter 5 review. Um, Hopefully it won't be too long. Um, I'll just go over everything that I think should, like all the good information that you guys should know. Won't be too in depth, but we'll have definitely some good information in it. So we'll start off with what we first learned, which was the monomers, polymers, and how we make and form bonds. So I already have a pre-made layout here. We're going to be talking about how we first break bonds, and that process is called dehydration synthesis. This is how we basically, or sorry, this is how we form bonds. The breaking of bonds would be hydrolysis, and we'll show you guys how to form and break the bond. So usually what would happen is we have an H and we have an OH. Let me um so that would just be H2O. So what would happen is there would be an enzyme. Let me use a different color. There will be an enzyme that takes this molecule, or it takes the monomers, breaks it, takes water out, and then forms a covalent bond. Now for each macromolecule, they have different names for the covalent bonds, but the type of bond is a covalent bond. So that's the gist of it forming. It basically takes two monomers, and this could be two monomers, or it could be, as right here it has basically the whole way of creating a monomer to polymer, or just extending onto a polymer. It uses an enzyme. It takes out water, and it forms a covalent bond by forcing them to bond, and it creates a covalent bond. Moving on to hydrolysis. This one is basically the opposite of dehydration synthesis. What you would need to do to break the bond is an enzyme. So how did I draw it? An enzyme. You will need to put water into the enzyme, and what would happen is the enzyme will basically take the water, it would force this bond to break, it will add a hydrogen onto this side, and then OH on this side, and that's how it breaks the bond. It uses an enzyme using a dehydration reaction, and sorry, not using a hydrolysis, it breaks it using an enzyme. It puts water into the system, which then puts hydrogen back onto the one monomer and OH back onto the second monomer. And that's basically dehydration synthesis and hydrolysis. Uh, Now talking about monomers and polymers, real quick. Monomer, I think monomer. A monomer, you can think about it, is the building blocks. The building blocks for polymers. Oops. And polymers are basically many monomers combined or connected. So a monomer would just be like this, just one, this would be one monomer, for example. And so this would be a monomer. And a polymer would just be more than one or more than one. This would be a polymer because it's more than one monomers. But it's made out of, this one would be made out of three monomers. So that's one monomer, two monomer, and three monomers that make that polymer. But the whole gist of it is a monomer is one, it's a building block for polymers, and a polymer is more than one monomers combined using, of course, dehydration synthesis. All right. Now that we talked about the main things that we need to know at the beginning, we can start talking about the four main different types of macromolecules. And the first one we're going to talk about is lipids. And everything you need to know about lipids. Okay. Now, lipids usually consist of two main. They don't contain a true monomer or a true polymer. The three main types that actually use monomers and polymers are carbohydrates, proteins, and nucleic acids, which we'll also go over. But lipids do not have true monomers, but what they have two main components. That's a three carbon alcohol, which is basically called, the name of that three carbon alcohol is called glycerol. And it also uses a carboxyl group followed by hydrocarbons. So it's going to be called it's going to be made out of a fatty acid. Actually, let me write. So hydrocarbons are basically just um, uh, a hydrogen and carbon like layout. So it'd be like hydrogen, carbon. That's a hydrocarbon, just hydrogen and just carbons. And a carboxyl group, again, since it's a fatty acid, you can remember acid as carboxyl group. So carboxyl group would be here OH double carbon C and NH. I think so. Let me double check. So it's actually, sorry, I had that wrong. I got confused. It's actually a C double bonded O and an OH. So basically C double bonded O and a C to an O and an H. So we can remember that as like the hydroxyl group and the carbonyl group, and then you just combine it into one, which makes the carboxyl group. And then hydrocarbons are again hydrogens and carbons, and that would make a fatty acid. Now, these two will combine. So you can have, for example, you have your 3 carbon alcohol here, and they do have hydrogens on them, but you have the whole point is you have your 3 carbon alcohol, then your fatty acids will connect to them. So you have your hydroxyl group. Now remember, this is 3 fatty acids. I'm pretty sure it connects to here, and it's just hydrocarbons after this, or also here. And you have these are they call them the hydrocarbon tails of fatty acids, and we'll talk about the saturated and unsaturated ones. But if you connect the glycerol with fatty acids, you have a bond here via, remember, dehydration synthesis, and all of these bonds are called ester bonds, but they're also covalent bonds. They're also covalent bonds, but they're also called ester bonds. And when you have 3 fatty acids on one glycerol, so this would be one glycerol and 3 fatty acids. You basically form something called a triglyceride. So tri meaning 3, glyceride. And this is what is called fat. This is basically what fat is a triglyceride. All right. So that's one thing of lipid. That's one type of lipid. So I'll put that off to the side. Now we can talk about the saturated and unsaturated. So saturated means it has all single bonds, single covalent bonds on the hydrocarbons. For example, you have a hydro and a carbon, and a hydro and a carbon. This would be considered a saturated hydrocarbon end, and an unsaturated would look something like this. It would have an H, C, and then it might have a double bond H, which creates a kink, and you could also have another one, for example, which would then create another kink. So when you have double bonds, so this one would have double bonds, one or more double bonds, then this is what would be an unsaturated hydrocarbon. This one would be solid at room temperature. At room temperature, I'm gonna write temp. This one would be liquid at room temp. And again, unsaturated has kinks, and this one has no kinks. Kinks meaning it bends right here. So this bend is a kink, this bend is a kink. The saturated would just mean it's straight. And that's all for saturated and unsaturated. Next thing we're going to talk about is phospholipid, which is again really important for your membrane, but this one we're just going to talk about the properties and how it's actually made. Phospholipid. It basically takes the 3-carbon glycerol. It then uses 1 carbon to create a phosphate head. So P and a bunch of oxygens. I think it was like that's a phosphate group, and carbons to create its two. We have 2 fatty acid groups. Now, this will also have a bunch of hydrocarbons down the line. Those are hydrocarbons. And what this will do, one property of phospholipids is this end right here, since it's hydrocarbons, and hydrocarbons are again nonpolar, which makes it hydrophobic. And we have this end, which is polar, and it's hydrophilic. So this means that this molecule has both hydrophilic and hydrophobic. And what we call this is amphipathic. I think that's how I spell it. Amphipathic. An amphipathic molecule meaning it's both hydrophilic and it's hydrophobic. And this is what allows the creation of the cellular membrane, the bilayer, of course. But we'll talk about that in chapter 6 and 7. And that is it for phospholipids. And one last thing for lipids specifically is you have a 4 fused ring plus maybe a chemical group. And this would equal a steroid, which is also, steroids are also hydrophobic. They're also hydrophobic. So basically what that would look like, is a, I do not know how to draw this. Bam, 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 bam. So you'll have that connected to another one. That is not supposed to be. And then you'll have another one right here. And then you'll have like a half one right here. and then your chemical group would be attached to this. So this would be your chemical group. So this is a steroid right here. And there's different types of steroids. For example, you have cholesterol. Cholesterol is a type of steroid that is also used in your membrane, which we'll also talk about in chapter 7. But all you have to know is that a chemical group or a steroid, which is 4-fused ring, is also considered a lipid. So that would be everything for a lipid. We will now move on to carbohydrates. Carbohydrates are pretty straightforward. The monomer. Now we're getting to monomers. So a carbohydrate has a monomer and a polymer. So the monomer is a monosaccharide and a polymer is a polysaccharide. I think it's two C's. There, I'd spelled it wrong. All right, now that we have the polymer, the monosaccharide and the polysaccharide as a monomer and a polymer, we know that a monosaccharide, multiple monosaccharides will make a polymer, and we'll talk about like types of them. So, for example, we have uh, we have three type we have different types of carbon sugars. So you can have a triose, triosis, which is equal to a 3 carbon sugar. So that will have like 3 carbons, and you could have, so a type of that would be glyceraldehyde. You'll have a pentose, which is a 5 carbon sugar. A type of that would be ribose. And you'll have a 6 carbon sugar, which is also called hexose. Which you could also call, or which an example would be glucose. So those are different types. You could have 3 carbon, 5 carbon, or 6 carbon, or triose, pentose, or hexose. Um, you could also have them each. You could have them as with a. You could have them. You could have them as aldehyde, which means the functional group or where the work is happening is towards the end. So, for example, right here it's happening towards the end, or like towards the top or bottom or towards the end of the molecule. Or if it's in the middle it's called ketose or ketosis, where, for example, it would be. This is an example. So it's right here in the middle where That's the functional group that where the work is happening is in the middle. So that's aldehyde and ketose. Um, forming forming wise, you can form it a disaccharide. So disaccharide means two two saccharides. So a disaccharide means two monomers basically combined. So if you combine a glucose molecule plus a glucose, you will get the disaccharide of maltose. And if you combine glucose plus fructose, you get the disaccharide of sucrose. So that's a disaccharide, but you can also have a full-on polymer. You can also have a full-on polymer, or sorry, polysaccharide, which is also considered like you can have starch, uh, or glucose. So you can what plants, for example, plants store polysaccharides as starch. So that's also really important to know. Oops. Plants store polysaccharides as starch. That's really important to know. And animals store as glycogen. So animals store polysaccharides as glycogen, and plants store them as starch. There's also So that's also really important to know. And then the last thing is structural polysaccharides, such as cellulose. So the the polysaccharide cellulose makes the cell wall. So that is the last thing you need to know about carbohydrates. So the monomer is monosaccharide, polymer polysaccharide. You can have a trisose, pentose, or hexose, which is the same thing as saying 3 carbon sugar, 5 carbon sugar, 6 carbon sugar. You can have an aldehyde, which means the function group is at the end, or ketose, which means the function group is in the middle. You can have a disaccharide, which is just two monosaccharides making a polysaccharide. And then we have to remember that plants store polysaccharides as starch, and animals store polysaccharides as glycogen. Uh, cellulose makes cellulose makes a cell wall in plants, not animals. It's only plants, because plants are the only ones that have a cell wall. That is a carbohydrate. So we can move on to the next one, which will be proteins. Proteins monomer is an amino acid. Polymer is a polypeptide. And they form form? They form a peptide bond. I think, did I talk about that? No, I did not. Let me correct one, not correct, but let me add one more information for that. These create a glycosidic bond. Carbohydrates create a glycosidic bond. I forgot to mention that. Proteins create a peptide bond. So you can just remember that by a polypeptide is the polymer, and the peptide bond is what actually forms the bonds. Now, we need to know the four structures of proteins. So we'll talk about from here. From a polypeptide, a polypeptide is a sequence of amino acids. Sequence of amino acids. And that is the same thing as saying that equals the same thing as saying primary structure. The sequence of amino acids is the exact same thing as saying primary structure. Now, the primary structure is basically amino acid. So the amino acid is a monomer, and there are 20 different types of amino acids. So amino acids there's 20 amino acids, and with them in different combinations, they equal the primary structure. They could be different, they could be, so the sequence would be the amino acid sequence, also you can call it, or primary structure. Now, from a primary structure, you want to go to a secondary structure, and a secondary structure is hydrogen bonds between amino acids. It's hydrogen bonds between amino acids, and that would equal secondary structure. Now, I'm just gonna. So the hydrogen bonds between amino acids equals the secondary structure. This would be considered folding the neighborhood, according to the professor. Folding the neighborhood just means like at different sections it creates a secondary structure. And the two types of secondary structure are alpha helix or beta pleated sheets. Those are the only two types that secondary structure creates, alpha helix and beta pleated sheets, using hydrogen bonds between the amino acids. Okay. After secondary structure, you have tertiary structure, which is also all bonds, all bonds possible. So every single bond is possible in tertiary structure. You have hydrogen bonds, you have covalent bonds, you have hydrophobic interactions, you have ionic bonds. Every single bond possible would be for tertiary structure equals tertiary structure. So anything is possible. That's what combines it and makes it into a 3D shape. So from tertiary structure, this would be a completed protein for some proteins. However, some of them go into quaternary structure. That's also saying all bonds besides covalent. Or you can say all non-covalent bonds. So all non-covalent bonds equals quaternary structure. And quaternary structure basically just means multiple tertiary structures combined. So that would be uh, multiple proteins coming into one, but remember that they all can split up accordingly. So that's why it's not covalent bonds, because covalent bonds are stronger than the rest. So it can break off easily. Once it's done with its job, it can break off, and it's just multiple completed proteins combined into one. Um, a quick recap of what, oops, a quick recap of what proteins do, and then we'll be done with proteins. Proteins do everything, basically. And I'm going to list some of them. Some of them considered as enzymes. Some of them are protective. For example, fighting off viruses and other bacteria. Storage. Some of them are receptors. Bro, my handwriting is not it today. Some of them are hormonal. Some of them are structural, like your muscle proteins, your muscles. Some of them are contractile and motor proteins. And that is some things that proteins do. But remember, proteins kind of do everything. So again, monomers, amino acid, they have 20 amino acids. And also let me write out the main sequence of an amino acid that you guys should know by now. You have your R group, that group, and you have your amino group. Or, sorry, I don't know why I put N. And your carboxyl group. This part, this part of your amino acids that part of your amino acids always stays the same in all amino acids. And the R group is what is changes in all the amino acids. So all amino acids have different R groups, but the rest of the amino acid is the same. And that's pretty much for proteins. So we will head over to the last one, which is nucleic acids. A little harder one, but we'll get through it. It is made up of three main components that make up the monomer. Three main things, three main make up the monomer. So remember, the three main things do not make up a polymer, but the three main things make up the monomer, and that main molecule would be the monomer that combines to each other to make the polymer. So we'll talk about how the monomer is made. The monomer has a 5 carbon sugar, or you can say it is a pentose. Next, you have your nitrogenous base. Then you have your phosphate group. Those are the three things that make up your monomer. Not the polymer, but the monomer. Now, for the nitrogenous base, there is stuff to know. So we'll do this. You have a purine, and you have a pyridamine. A pyrimidine. Pyrimidine. The nitrogenous base has a purine and a pyrimidine, and we came up with the way to remember how or what to name it is pure as gold. So pure starting with purine, so it would be pure as gold meaning adenine, and gold meaning guanine. And then the rest are pyrimidines. So that's how you remember. You remember pure as gold. The only two that are purines are adenine and guanine, and the rest are pyrimidines. So one would be cytosine, one would be thymine, and the other one would be uracil. Now, that is the monomer. It's made out of a 5 carbon sugar, nitrogenous base, and a phosphate group. So this is a 5 carbon sugar. You have your one prime right here. This is a prime, one prime. Oops. Two prime, three prime, fourth prime, and fifth prime. Yeah, basically, because that's five carbons, so you have, they call them prime. So one, two, three, four, and fifth prime. The second prime, or excuse me, the first prime connects or has the phosphate group. Okay, and the five prime, or excuse me, sorry about that. The first prime actually has the nitrogenous base, and the five prime has the phosphate group. So that's how it's connected. You have your five carbon sugar, which has a nitrogenous base connected to the one prime, and a phosphate group connected to the five prime. Now, this would be your monomer, so of course we need to make it a polymer. The polymer, so what this is called is the monomer, forgot to name them. The monomer equals a nucleotide. The polymer would be a polynucleotide. So now we need to make a polynucleotide because this structure right here would be your nucleotide. So this would be a nucleotide. Now we need to make a polynucleotide. So how that happens is you have different types of polynucleotides, which we'll break up right now, actually. We'll do smaller. We have our monomer, which we'll just take right here. We have DNA, and you have RNA, which we'll break up. So do two different types of polymers or polynucleotides. You have DNA and RNA, and they have somewhat different structures. So the nucleotide starts off with the 5 prime and goes down to a 3 prime. The 5 prime always connects to the 3 prime. So what I mean by that is the phosphate group. So if we had a polynucleotide, if this was our polynucleotide, for example, this side would be the 5 prime, this side will be 3 prime, and we'll have another polynucleotide which will have a 5 prime and a 3 prime. And the 5 prime will always connect to the 3 prime. The 3 prime never connects to the 5 prime. So what I mean by that is like the 5 joins to the 3, and we'll have another polynucleotide right here with the 5 prime and a 3 prime, and the 5 prime will again connect to the 3. So it goes from top to bottom in an organized manner. The bond, the bond that these two create equals a phosphodiester bond. The bond is a phosphodiester bond. So the bond between the phosphate group and the 3 prime is a phosphodiester bond. DNA. The main difference between DNA and RNA, or there's a couple main differences. DNA is double-stranded. The double-stranded helix, because it's a helix. It's also anti-parallel. Anti-parallel means you'll have a 5 prime going down to a 3 prime. That's one strand again, but it's a double strand. So that means you'll have a 3 prime. And remember, 5 prime always connects to 3 prime. A 5 prime going that way. So this right here is anti-parallel, where the 5 is with the 3 and 3 is with the 5. So it's anti-parallel, going in opposite directions. The double-stranded helix, that is. Now, the nitrogenous bases, DNA only has A, T, C, and G. It does not have U. Remember, we actually have 5. We have adenine, guanine, cytosine, thymine, and uracil. That's 5. But DNA has 4 and RNA has 4. DNA goes with T, and they always go with T. It does not go with C or G or U for DNA. C always goes with G. So for example, if I had A, C, G, A, T. And if this is a 5 prime right here, if this is the 5 prime end, this is 3 prime, this would be 3 prime right here, and this would be 5 prime. A will go with T, C will go with G, G will go with C, A with T, A with T, T with A. So adenine is adenine with thymine, cytosine with guanine, guanine with cytosine, and it just keeps going down in an antiparallel matter. The bonds between them, these bonds right here, are hydrogen bonds. Those are hydrogen bonds. And the last main thing is on the 5 carbon sugar where there's a 5 prime here, or sorry, a 1 prime, prime. The second prime right here determines whether it's a hydrogen, no, sorry, if it's a DNA or RNA. Deoxy, DNA stands for deoxyribonucleic acid. Meaning the deoxy means it's removing an oxygen. So in the second prime, to know, it will have a carbon. It will have a carbon at the second prime. And instead of it having OH, this is wrong because what would happen is it's deoxy. So instead of it having an OH, it will just have an H. So deoxy, meaning it's removing this oxygen right here. However, on RNA, on RNA, the second prime would look like something like this. It would have H, C, and O, and then it would be connected to an OH. So it has that oxygen at the second prime. At this second prime right here, it will have, it would either have no oxygen, which means it's DNA, deoxy, or it will have oxygen, which means it's RNA. And then, of course, for RNA, RNA is going to be single-stranded, single strand. It's going to have, of course, the oxygen at the second prime, and the nitrogen bases are going to be like follow: A with U and C with G. So the only thing that changes is the T. RNA does not have the T, but it has the uracil or U, and it always goes with A, and C with G. So C with G stays the same for RNA and DNA. The only thing that changes is DNA has thymine, which is the T, and RNA has uracil, which is the U, and they both connect to the A. So that would be it for Chapter 5 review. We hit, again, what are monomers, what are polymers. We hit the 4 types of macromolecules. We hit how do the bonds form and break. And we hit lipids, carbohydrates, proteins, and nucleic acids, and how they are formed and what they do. So if you have any other questions, just let me know.